When Alzheimer's disease moves through the architecture of the brain, it dismantles the living tissue in a systematic, regional order. In 1980, scientists identified a massive drop in a specific peptide called somatostatin. It was the first major chemical deficit found in the Alzheimer's cortex that didn't involve acetylcholine. For the next 4 decades, the field treated this loss as a secondary consequence of the illness. The assumption was that the cells producing somatostatin were simply dying alongside everything else in the late stages of tissue collapse. This chart maps the timeline of the disease using data from the 2024 Seattle Alzheimer's Disease Brain Cell Atlas. It plots human tissue samples along a pseudo-progression axis, dividing the disease into an early, cognitively silent preclinical phase and a late degenerative phase. If we plot the survival of the somatostatin-producing interneurons on this axis, they don't die late. They vanish entirely within the early, silent window, and they vanish alone. Excitatory neurons, parvalbumin interneurons, and even the classic neurofibrillary tangles remain largely absent during this same period. The other major cell classes only begin to collapse much later. Because these cells die before the surrounding architecture fails, their loss is not a symptom of the disease's late stage. It is the trigger that initiates the compounding failure of the circuit. To understand why this specific cell's death is so catastrophic, we have to look at how it is wired. Most inhibitory interneurons act as a simple brake. They receive an external signal and clamp down to stop a neural circuit from firing. The somatostatin cell is different. It functions as a mechanical governor, an engineering device driven by the very speed it is designed to limit. The somatostatin cell receives its excitatory input from the exact pyramidal cells it is responsible for inhibiting. It applies feedback inhibition to the distal dendrites of those same pyramidal cells. Instead of merely controlling the timing of a spike, it dictates exactly how much the target cell is allowed to fire. When a simple brake breaks, an engine is merely unguarded. When a governor breaks, the fault compounds. As the uninhibited neurons fire faster, the system that was supposed to restrain them is actively degrading, making it progressively less able to slow them down. This governor regulates its circuit by applying 2 distinct restraints simultaneously. First is the electrical restraint. The cell releases the somatostatin peptide onto the pyramidal neuron. This augments the M current, a voltage-dependent potassium channel that makes the target neuron less willing to fire repetitively. It acts as a tonic, dampening influence on the local network. The second restraint is entirely biochemical, and it controls the brain's waste disposal. At the cellular membrane, the somatostatin peptide binds to specific receptor subtypes, SST1 and SST4. This binding sets off a defined cascade. The receptors trigger a regulatory molecule called alpha-endosulfine, which then interacts with a specific ATP-sensitive potassium channel. That interaction directly regulates the activity of a large enzyme called neprilysin. Neprilysin is the principal protease in the brain responsible for degrading amyloid beta 42, the exact peptide that accumulates to form the plaques defining Alzheimer's disease. The somatostatin interneuron is operating 2 critical controls at once. It prevents the circuit from overheating electrically, and it physically licenses the enzymatic disposal of the poison that causes the illness. This raises a structural paradox. The somatostatin cell and its sibling, the parvalbumin interneuron, reside in the same tissue and face the same amyloid burden, yet the somatostatin cell dies years earlier. The difference lies in a structure called the perineuronal net. This is a condensed lattice of sulfated sugars that wraps tightly around certain neurons, functioning as a physical barrier. Parvalbumin cells are heavily ensheathed in this net. For the brain's immune cells, microglia, to attack a parvalbumin neuron, they must first expend time and energy enzymatically degrading that lattice. The vast majority of somatostatin cells carry no perineuronal net. They are built without armor. In early-stage Alzheimer's, hyperactive microglia run a specific elimination program that preferentially targets and prunes inhibitory synapses. Because the somatostatin cell lacks a protective net, it presents no latency step. Microglia can immediately engulf and dismantle the cell's synaptic connections without delay. The architecture that allows the somatostatin cell to be highly plastic also leaves it totally exposed to an inflammatory immune response. The consequences of this vulnerability concentrate in one specific anatomical layer of the hippocampus. This is a cross-section of the hippocampus. If we isolate the CA1 region, we find a microscopic lamina called the stratum lacunosum-moleculare. This specific layer is the exact terminal point where long-range memory inputs from the entorhinal cortex arrive to pass their signal into the hippocampus. It is also the physical territory where the local somatostatin cell sends its axon to apply its inhibitory brake onto those incoming memory signals. And structurally, the neprilysin capacity that depends on somatostatin receptors is heavily localized to this identical thin layer. For decades, the circuit physiologists mapping these memory pathways and the biochemists studying amyloid clearance published parallel findings without overlapping. Placing the data side by side reveals a ruthless biological economy. A single microglial assault on an unnetted cell in the specific layer achieves two things at once. It severs the primary memory gateway to the hippocampus and shuts off the local disposal system for amyloid. Because this specific cell governs both electrical excitability and biological waste clearance, its sudden death triggers a catastrophic self-amplifying feedback loop. Biochemically, rising amyloid injures the somatostatin cell, lowering neprilysin activity and causing amyloid to accumulate. Electrically, the lost cell lifts the inhibitory brake, leaving pyramidal neurons to fire erratically. Since amyloid beta is released during firing, these hyperactive neurons actively secrete even more amyloid. The failure of the governor drives the pathology from both ends. It shuts down the clearance of the poison, while simultaneously forcing the surrounding tissue to produce more of it. There is an assumption that because spatial navigation fails early in Alzheimer's disease, the early loss of somatostatin cells must be the direct cause of the breakdown in the brain's spatial grid cells. Anatomically, this is impossible. This diagram of the entorhinal cortex shows grid cells reside exclusively in layer 2, a territory controlled by parvalbumin interneurons. Somatostatin axons don't reach layer 2. They are strictly confined to regulating the deep output pathways of layers 3, 4, and 5. The grid cells fail later because the loss of the deep layer governor creates a chronically toxic, hyperexcitable environment that eventually kills the surrounding tissue. It is a secondary consequence, not a primary computational failure. This mechanism tells us exactly why blunt interventions fail. Attempting to suppress hyperactive somatostatin cells to calm the electrical network only accelerates the biochemical loop, because silencing the cell stops it from releasing the peptide required for amyloid clearance. The therapeutic requirement is to intervene early, in the silent preclinical window, using receptor agonists that mimic the peptide. By acting directly on the SST4 receptors, we can bypass the dying cell to manually restore both the electrical brake and the clearance license. We have to repair the governor before the engine destroys itself.